Hello, and thank you for your interest in my poster titled An Examination of the Effects of Body Temperature on QT Interval in Non-Naive Telemetered Göttingen Minipigs, presented at the ACT Conference in Austin, Texas, 2024. My name is Miri Pannu, Associate Scientific Director of Safety Pharmacology at Altasciences, and I will give you a brief overview of the work we've conducted. Body temperature changes have been shown to affect the QT-QTCH interval. Given the known inverse relationship in other species, the study objective was designed to propose a correction formula for routine application in miniature swine. 8 male telemetered Göttingen Minipigs were used to determine the relationship between QT-QTCH interval and body temperature. The study was conducted in 2 phases. In Phase 1, animals received a singular IM injection of xylitol. In Phase 2, animals were anesthetized with isoflurane, intubated to provide consistent oxygen levels, and maintained in a constant anesthetic plane with isoflurane. At least 10 minutes of continuous readings were collected at normal body temperature. Body temperature was cooled to a target of 34 degrees Celsius and then warmed to a target at 41 degrees Celsius. The resulting data was analyzed post-acquisition on a validated system with EMCA ECG Auto. As you can see here in Figure 1, significant increases in QT and QTCH interval were observed following administration of xylitol. Peak difference in covariant adjusted mean for QT and QTCH occurred at 2 hours post-dose, with values returning to normal by approximately 4 hours post-dose. Here in Figure 2, significant decreases in body temperature were observed following administration of xylitol. Peak difference in covariant adjusted mean occurred at 2 hours post-dose, with values returning to normal by approximately 4 hours post-dose. Given the known suppression of thermoregulation following administration of injectable anesthesia, these changes were to be expected. The duration of change was consistent with the known PK properties of xylitol. In conclusion, xylitol administered via IM injection at 9 mg/kg produced expected physiological changes, further outlining the relationship between body temperature and QTCH interval in Göttingen minipigs in both conscious, as seen in Phase 1, and anesthetized, as seen in Phase 2, states. The study provided evidence of an inverse apparent linear relationship between the 2 parameters. A QTCH formula with a correction factor for body temperature changes, QT-CHCT, was derived for both conscious and anesthetized states.